Our next presenter is KaileeReed. Kailee is a third-year PhD student in cell and molecular biology. And with their degree, they hope to become a data scientist and propel RNA biology forward with the use of computational techniques. The title of Kailee's presentation is Identifying Mechanisms of Primary MicroRNA Biogenesis.
DNA, as we all know, is the genetic material of all organisms on Earth, and it is divided into functional units known as genes. In order for a protein to be made, there is an intermediate step known as messenger RNA, and these are the messages or instructions for a product, a protein. The transcription and translation processes are both highly regulated, and one of these potential regulators are called microRNAs. These are just small RNAs that bind to multiple targets or genes that can ultimately affect protein production. So instead of controlling one single gene, microRNAs can control the entire pathway, making them a very powerful regulator. They are so powerful that they have been shown to be involved in essentially every biological process, for example, in various cancers, development, and even infertility. But how do these microRNAs become mature enough to be switches that control when a protein is produced or not produced? There's a protein complex that you can kind of think as a gatekeeper that scans the genome looking for these microRNA structures. And once they're recognized, this complex helps start the process of a primary microRNA becoming a mature microRNA. However, the mechanism of how this gatekeeper recognizes and cleaves a primary microRNA is poorly understood in most species. My research focuses on how early or primary microRNAs are recognized and processed to become mature microRNAs. Surprisingly, you and a tiny worm called C. elegans have quite a bit in common in terms of genetics. Small RNAs, like microRNAs, are highly conserved between us and worms. So in our lab, we utilize them to study small RNA pathways. In our preliminary data, we used DNA sequencing technologies and found that there are other players that help the gatekeeper recognize primary microRNAs in worms, which could possibly extend to other organisms due to their high conservation. This research is uncovering complex mechanisms behind the microRNA production pathway, which is leading to understanding complex diseases and their mechanisms. Thank you.